Okay, hello everybody. Welcome to our webinar, hosted here by Novolyze, Smart Food Safety and Quality, the Acheson Group, and Food Safety, Microbiology and Hygiene. Our topic today is smart environmental monitoring, controlling pathogens and preventing outbreaks. And I am Ashley Diamond with Novolyze. We've got a great presentation lined up for you today. First of all, some housekeeping. We welcome your questions. Please use the questions panel to submit all questions, and we'll go ahead and answer those at the end of the webinar. If you have a question after the webinar, please also feel free to reach out at marketing@novolyze.com, or you can use the Contact Us form on the novolyze.com website. If you're having any trouble, please try logging out and logging back in. There are also alternative methods to join. You can use the link given on the screen there and the webinar ID in your confirmation email. You can also join via phone. Details on that are given in your confirmation email. This webinar is being recorded, and you will receive a link to the recording after the webinar is complete. And that is that. So I'd like to introduce our presenters and moderator here. First of all, we have Dr. David Acheson, who has more than 40 years of medical and food safety experience, having practiced medicine for several years and serving in prominent roles with both the US Food and Drug Administration, FDA, and USDA Food Safety and Inspection Service prior to forming the Acheson Group in 2013. At the USDA, David served as the Chief Medical Officer of the Food Safety and Inspection Service. As the FDA's Associate Commissioner for Foods, he held agency-wide leadership for all food and feed issues, and he developed the 2007 Food Protection Plan, which served as the basis for many of the authorities granted to the FDA by the Food Safety Modernization Act. Welcome, David. We have next Francois Brodachon, who is a trained microbiologist from Pasteur Institute in Paris, France, and the Università Cattolica del Sacro Cuore in Piacenza, Italy. With over 20 years of experience, he has worked since 2017 as a food safety consultant for the industry, with a focus on dairy, infant nutrition, and confectionery. He specializes in crisis and technical support, microbial risk management, microbial risk assessment, food safety and hygiene, fermentation, and biopreservation. He is the principal consultant for food safety, microbiology, and hygiene, which has almost 100,000 followers on LinkedIn. And then our moderator is Novolyze's Karim F. Kanoush. He is founder and CEO at Novolyze. He is a food science engineer and also holds a master's degree in business development. Karim was previously with ETIA Ecotechnologies, where he took charge of their food division development, including the launch and development of this safe sterile technology for low moisture food pasteurization. Karim founded Novolyze in 2012 to help build a better, safer future for food at global scale, offering leading edge environmental monitoring and other food safety solutions, such as smart thermal process control and surrogate for process validation. And with that, I will pass it over to our moderator, Karim, and I hope you all enjoy the webinar.
Yeah, thank you, Ashley, and welcome everyone. So we have two fantastic panelists today, and we're going to speak about environmental monitoring and how digitizing can make it smarter. So before we kick it off, how about quick basic question on what is an environmental monitoring plan, and what's your perspective from both European and US side, since Francois, you're based in Europe, working mostly with European companies, also with global footprints, and David in the US with global company with European footprint. But there are some subtleties there, and I find it particularly interesting to have both point of view today. So how about you kick it off, David?
Yep, just getting off mute. Absolutely. No, Kareem, pleasure to join you today. Thank you. Well, in essence, to me, an environmental monitoring plan is a fairly simple definition, is that it's a verification tool to show that your environmental controls are working. It is not a tool that one uses to rely on controlling the risk per se. One needs multiple controls to control the risk in an environment, especially a post-processing environment, where there's concerns about a number of microbes getting into product after it's been processed and before it's been packaged. Those controls are multiple, and we can get into those as time allows. But the monitoring plan is all about verifying, my view, is verifying that those controls are working. And obviously, from a US perspective, any FDA registered facility that is under the Food Safety Modernization Act and making a ready-to-eat food is going to be required to have an environmental monitoring plan if they have product exposed to the environment. So in a nutshell, that's what it is. It's a verification tool.
Yeah, thanks, David. And Francois, what's the perspective you have from France, and how do you see the European, at least France and European in general, environmental monitoring plan being different from the US standard and also the US expectation for those who are exporting to the US?
The first point is I'm fully supporting the position of David mentioning that environmental monitoring plan is a proactive approach to anticipate failure, because per se there is this bad habit to believe that finished product testing does help, but we know it's only providing a reactive approach. If you fail on your finished product, you need to destroy everything. Now, if you fail an environmental monitoring program, it helps you finding by root cause analysis where you have other issue in zoning, in cleaning practice, in coding. So per se, it's not something that will makes you better, but it will tell you how good or how bad you are. In Europe, it's a no-brainer. It's legally binding in EU 2073 regulation since 2005. So you have to do it for Listeria monocytogenes for ready-to-eat foods. And if you look in the documents, you have to do it as well for Salmonella in low moisture food. And basically the only difference is that we don't have clear guidelines from the authorities, apart one guide from the European Reference Laboratory for Listeria in France. ANSES is providing a guideline, but it's already from 2012. But apart from this, it's based on your own assessment. And most of the companies are discovering still in 2023 processing environment monitoring, and they rely on guidelines, and most of them come from the US. We have some guidelines in Europe, very recent one, but basically most of the guidelines, they are US driven.
And you mentioned Listeria and Salmonella. Is it specifically these two bugs which are triggered by the ANSES, or is it more widely?
You got a third one, which is making the news presently for infant production for phase 1, or which is Chronobacter spp. Basically, when you look in the environment, we mentioned the pathogen of concern, which are Listeria monocytogenes. It's very straightforward on the FDA website. Wet and chilled, it's Listeria monocytogenes. Dry and hot, that's Salmonella spp. Infant food, it's Chronobacter spp. And that's the 3 pathogens of concern you have to be looking for. And in terms of hygiene indicators, Listeria spp is helping for Listeria monocytogenes, and Enterobacteriaceae is helping only for hygiene. This is an important point. In the EU regulation, in the update from 27, it's mentioned that there is no correlation in terms of environment monitoring between Salmonella and Enterobacteriaceae. They don't provide the same information. And it's up to the food business operators to prove there is a correlation between Chronobacter and Enterobacteriaceae, but nowadays it's not been updated in the regulation. But nowadays we know there is no correlation per se, and that's something we're going to discuss later with the issue with resident strain.
David, you want to comment on that? So you were
no, absolutely. Totally aligned with what Francois is saying. Is those are the 3 bugs of concern, and I think likely everybody on the call is very familiar with controlling risks of Salmonella and Listeria in the environment. Certainly a very hot topic and a huge area of focus is Chronobacter sakazaki and its relation to infant formula. You know, and I think, you know, in the interests of being current and topical, right now we've got requests from FDA for infant formula manufacturers in the US to inform them when they have findings of Chronobacter sakazaki in finished product, even though it's still under their control. That's so it's good timing for this topic because it's always been an issue of environmental. I think since Food Safety Modernization Act put in a regulatory requirement for this under those conditions that I outlined of ready to eat, that's continuing to evolve, and there's now an approach from the FDA to ask for new regulatory authorities in relation to infant formula and foods for infants, which is also heavily focused on knowing what's in your environment. You know, and I think we may get into this more, but I'll say it up front, is there is a perception, Kareem, that, or there used to be, and I think it still is to some extent, is that, yeah, I've got an environmental monitoring program and I never find anything, therefore I must be really good. And I think that is 100% incorrect. It's like, yeah, you need to find stuff because it doesn't matter how good your facility is in a food plant, you have got pathogens lurking in zone 3s for sure. And if you're not finding those, they're probably sneaking around without you knowing where they are, and they'll be on zone 2s and then in finished product before you know it. So, you know, a fundamental component to an environmental monitoring plan is the concept of seek and destroy. You want to find it. You don't want to not find it.
Yeah, it never feels good to find it, but I think it's good to remind always that if you find it, you did a good job, and there is no one out there who doesn't have any bugs there. I mean, the best in class, they have pathogens, and you need to find them to make sure that your program is working. And I think we never repeat that enough to make everyone feel good and not feel guilty when they find one of those bugs.
No, right. And there, I mean, I think there's a fear. Well, if FDA comes in and sees that I've found them, are they going to ding me? And the answer is no. You know, if you find them and you don't act on them, then for sure, I mean, you're setting yourself up for a problem. So the environmental monitoring plan, as I said, is a verification step, but you've got to follow through. And when you find it, you've got to take the appropriate corrective actions, do a root cause follow up. So it's not simply, oh, found it, I cleaned and I moved on. It's like, you got to be thinking about this constantly. And I would say the environment, the risk environment in this space is the hottest I've ever seen it. I mean, it is absolutely a space that if you're not on top of, you will be heading for trouble with the regulators.
And I think you pointed out a very important point here is you need to find it, and then you need to make sure you did, and to follow up and be able to prove you did something. And this is where sometimes stuff falls in the crack, because there's just so much going on in the factory, and being able to follow on all these actions and getting the traceability that's been taken care of can be a bit cumbersome, and that's where tools can be helpful. I think we should go on the next slide, and out there on that one, we got some interesting outbreak. You started to, I mean, you guys both commented on the infant formula issue with Chronobacter. We had similar issues with Salmonella in Europe, and this comes regularly. Anything you want to add on this one, David? And before I turn to Francois, we got pretty big cases in France over the last few years.
Well, this is just sadly a short list of, it's certainly not comprehensive, of where we've seen notable recalls and outbreaks linked to environmental contamination. And the obvious one, that right up there, peanut products. The poster child for this, is I think probably everybody on the call is aware, is what happened with Peanut Corporation of America, which triggered a whole cascade of requirements and changes, and you know, it drove other aspects of regulatory strategy in the United States. But it is the most critical thing that drove the requirement in the Food Safety Modernization Act, because the timing of that outbreak coincided with the creation of the statutes around the Food Safety Modernization Act. So there was a reason for it, there was a trigger for it. But each one of those on the list there are obviously, they're significant. And as I say, this is far from a complete list. But the other way to flip this around, Kareem, is is this just a cost center and a regulatory box to check? It certainly is both of those two things. It does cost you, and it is a regulatory box. But as these examples illustrate, when your environment goes wrong and you have a contamination issue, you're looking at an extraordinarily costly event potentially, both in terms of direct recall costs and indirect reputational costs when something goes wrong. So it's an investment that one absolutely has to make in today's public health and regulatory environment in the US for sure. And I think it's worldwide, frankly. I think love to get the European perspective on that, but it's important.
Well, it's a cost just like soap to wash our hands and wash the equipment. It's a cost center, but no one would think about not using soap or detergent to clean their hands and their equipment. Francois, any insight on that?
Yeah, to come back on what David was mentioning, and processing environment monitoring as a cost, definitely, but not doing it is even more costly. So it's part of the job that you have to do. It's part of the return of the investment you need to do to ensure food safety. We mentioned here in this list quite important outbreaks which were significant in the US, but you need to have in mind that ICMSF started publication on the topic of processing and environment monitoring more than 25 years ago, but that was as the initial step. It's been known for a while by the food industry with internal guidelines, and it was a bit learning by doing inside different companies, aware or not of this situation. Personally, when I started in the cheese industry some 23 years ago, we had internal monitoring for already 10 years before I arrived. As soon as we made the identification that listeria was an ecological issue, not only coming from the milk, but also coming from the environment. So we had recently major outbreaks that really triggered the point also for authorities to be aware that this is a major issue, because you mentioned here some episodes that you had in the US. We had major one recently in Europe and also pre-COVID, and it was also a shock for the authority to discover that you could find listeria or you could find salmonella surviving in a factory for 5, 10, 15, 20 years. And I think nobody now today can say is not aware of the topic. And as David was mentioning, right now is a good moment to push for the existing guidelines. You've got old one from the Edmund Board of California, but it's from 2010, but it's already completely valid. You've got the same one for United Fresh Produce, but it's completely valid. So you have a lot of documentation out there. What is important is to make sure that it will not solve everything. It's not because suddenly you make swabs that you will solve the issue. If you make swabs, you will find something. What is important is what you do with the results, because if you find something, say, oh, listeria again, and you don't do anything about it, you're in big trouble. The point is a bit schizophrenic, because you're going to look for the pathogen. You don't want to find it, but you're still going to do your best to find it. And one of the points that was mentioned in the outbreaks of listeria or cantaloupe that triggered the guidelines for United Fresh Produce is that there is no incentive for the people who find. If you find listeria, you blame. Why did you find it? So it's something that all the authorities and the corporate structure needs to have in mind. If you have a quiet factory and you don't find anything in your factory, this is not good. What is good is having a factory that finds and comes with an action plan and propose preventive actions to solve the issue. And solving the issue is not cleaning and saying, hey, I clean, it's fine. No, it's really understanding what happened, where does it come from, and having in mind where you see the bug is not necessarily where the bug comes from.
Right.
Yeah, we got a couple of questions here. So we have one that we received in Spanish, actually, and that Ashley translated for us. So let me read it out loud. And I work in a frozen berry and frozen asparagus plant. Now at my plant, we do not have an internal microbiology lab, and we use an external lab, but we must send the samples by mail, which takes about 12 hours. Is that why I never find a positive sample?
Okay,
it shouldn't
it
be the
depends
reason.
how you send. No, it shouldn't be the reason. There are guides. We have an ISO standard, the the 18593 from June 2018 that provides the time from the swabbing unto the analysis that they should be stored at 4 degrees before they are analyzed, like any micro sample anyway. You've got guidelines that mention that samples should be sent chilled for analysis. The point is, there were recent studies from ANSES mentioning that chilling might link to false negative because the bugs are stressed, and info chilling might not be also the best option. What is important is, if you don't find anything, as David mentioning, maybe you're not looking where you should, don't blame the lab directly. Could be the lab, nevertheless. But the point is, it's challenging your environmental monitoring program. Most of the time, this is done internally. It's good to have a fresh pair of eyes. So either you take someone from the group, if you're part of a group from another factory that can help, or you can also, it depends, or you are good with the authorities, but it's good to get approval from the authorities of your environmental monitoring program, so they can tell you if it makes sense or not. But not finding anything, if surprisingly, yes, that's possible. If you only do zone 2, eventually zone 1, yeah, and you don't find anything, it's an option. The timing also is very important. I've seen some factories sometimes saying that they were doing processing environment monitoring, but they were making the swab after cleaning. This is not environmental monitoring, this is cleaning monitoring. So if you make the swab after cleaning, you're not looking for the environment, you're looking for the efficacy of the cleaning practices. And most of the guidelines that you find are clear with this point. The swabbing must occur 4 hours after the start of the shift or at the end of the shift before cleaning, but never after cleaning. So if you don't find anything and you anticipate you should find something, you can challenge the practice. The methods could be an option, but there could be other ones.
And any idea on how we can help this person, apart from swabbing better in different places, to challenge the lab, maybe some on purpose contamination of the swab to test the efficacy of the lab?
No, I mean, first is, if you work with an external lab, is knowing what's the experience of the lab in terms of looking for environmental swabbing. There is a new standard available, the ISO 16140.3, for the verification of the method. And if you're working with an accredited laboratory, and I hope this is what you're doing, he must ensure you that he has the capacity to do the testing. Then there could be indeed an issue with your swabs. There could be also an issue with the timing and the location of your swabs. I don't know how the points were decided, but an approach which I do like, but it's expensive, it's the swabathon, which the FDA is doing. They're doing a hell of a lot of sample in different zoning, and then based on the results, most of the time they're pretty good and they found isolates, and then they can define what would be the best points to look for, the most relevant one, and so on. But if you never find anything, like we said earlier, yeah, I will challenge that.
And what do you think, David? Anything to add on this one? Not a fan on proficiency testing either?
Well, I think we're aligned totally that you expect to find something, and if you're constantly seeing negatives, are you looking in the right place? Are you using the right transport? Does the lab know what they're doing? And these are all things to check into. And getting some outside help to look at that program overall is probably a good idea. I think the notion of trying to deliberately contaminate a swab with a known pathogen in a production environment is probably not the way I would recommend you go. In fact, I would definitively say don't do that. But no, I think obviously that doesn't sound like something's quite right. So it needs a deeper look.
All right, good. Thanks. I hope that will be helpful for the person who asked that question. So if we go to the next slide, please, Ashley, maybe a last word on the difference between the EMP programs in US and EU, because it seems like you two are very aligned on how it's run, and there is no critical difference here on how we're supposed to run them. I think in the zoning, there's maybe a different wording or a consideration there. Anyone wants to jump on this one?
The Codex Alimentarius only defines food contact and non-food contact surface, which basically for the US will be zone 1 on one side and zone 2 and zone 3 on the other. because zone 4 is outside production. In Europe, I will be a little bit sarcastic, but recent guidelines mentioned that you need to look for critical sample points. Of course, we were not wanted to look for uncritical sample points, but what are critical sample points? And that's based on your assessment. So the guidelines, they should be updated. The guidelines were in France, they're a bit delayed, but I hope that to get a recent update soon, a fourth version. It's not descriptive enough, and it's not very helping, which means that basically this guideline from Profel on Listeria monocytogenes guideline from November 2020 followed the Grignard episode in Hungary, where they found Listeria in chilling tunnel in frozen vegetable factory, which means that, coming to the questions you had before for frozen berries, you expect, if you have chilling tunnel, you expect Listeria. That's a given point somewhere. We don't have the proximity approach, and this is a bit of an issue because basically the proximity is often sometimes which triggers different actions depending if it's a zone 2, zone 3, or zone 1 is a given point. Zone 1 means the product is contaminated, but zone 2 means you need to go for reinforced testing, while zone 3 means you need to have a look if it went up to zone 2. We don't have this approach yet. Most of the companies in Europe working in infant production have the approach still on place, but for ready-to-eat food, we don't have it, and it's a bit of concern.
Yeah, thanks, Francois. How about we go to the next slide, Ashley, because see where time is running, and we had more open question here on how to run an EMP efficiently, and a couple of items there, like we say here, avoiding manual coordinations internally and in between sites, basically not misinformation, avoiding decentralized creation of identifiers, avoiding spreadsheets, avoiding manual CAPA generation. David, what does that inspire you from your experience and how you saw the systems evolving over the last few years?
I think I mean, to my earlier point, Karim, establishing a robust environmental monitoring program is a critical part of overall risk control, and it is the verification step, as I've said. In order to do that, one needs to have a very robust understanding of where to look in your plant. You know, and many facilities just create a very long list of potential places, and they swab a percentage of those. There's an upside and a downside to that. One can get complacent, and I prefer, yes, there's some routine sites that perhaps historically have caused you a problem and you want to keep checking up on them. But I love a program that's got a little bit of randomness to it. So the person doing the swabbing has got the mentality and the thought process, oh, that's a new crack. I haven't seen that hole before. We got a little water there. It's not on my list, but I'm going to go sample it. So it's sort of getting the person who's actually doing the sampling to understand the concept around seek and destroy. Now, with everything in food and technology, Karim, digitalizing data and automating it so that the information is captured, because much of what we do in food safety, thankfully, we get negative results, we get normality. But we need to track all of that so we're truly able to trend. And then when you get a hit, whether it's a zone 3 or a 2 or hopefully not a 1, one can take a corrective action and document that, again, ideally electronically in some way or another, so you have the record. You know, it's a classic paradigm. If you didn't write it down, you didn't do it. So I think leveraging, moving away from manual process across the whole food industry is just a smart way to go, because it's we're very data rich, but sometimes that doesn't make us information savvy. We need to have actionable information, and leveraging automation and digital technology to turn the myriad of quote normal stuff and extracting those key factors. You know, a big part of this too is the value of maps, and I love visual images of knowing where have I tested, what have I found, where am I seeing hits. I think it's a very helpful way, which obviously requires digitalization and leveraging technology as opposed to just looking Looking at an Excel spreadsheet with the name of where you took the sample, that's hard.
What do you think, Francois, on that one?
Well, it's a bit anticipating one of the next slides, but what is important is when you test on finished product and Rosie, you got old and release, so you test on your product, you have the time to go back to the product destroyed, so time to react. On the environment, if you have a positive swab, you need to say, okay, something went wrong. When was the last time I had a negative, I had a non-detectable result? Over this period, I need to investigate what's been going on. By the time I've got this positive, I need to come back and make some starburst sampling all around him, clean, and then do the same testing again. So if you do things properly, very fast, you end up with a huge amount of data, knowing that right now in France, you need to inform the authorities about the positives and your action plan, which means that if you come to the authorities with all a set of sheets and here, have a look, I've got an issue, you might end up closing your factory. You don't want that. Now, if you come with a clear story, clearly defined, digitalized, and you can provide this within 3-4 days, that's great. I mean, having in mind that most of the time we're supposed to do mock recalls, and we say that when you have mock recalls in less 2-4 hours, we have the product back, and most of the time in 2-4 hours, we don't have everything, and we fail most of the mock recall because of traceability. You don't want to do that as an environmental monitoring program. We've got a lot of information that Industry 4.0 is coming, Food Safety 4.0 is coming. What does it mean? It means that you have a lot of information to deal with. You cannot trust ChatGPT to do the work. You need to trust yourself, and you need to have the software that will gather everything. I will take a quote from Martin Widman from Cornell University, which I really like. What is important is not the time to result, it's the time to decision. We've been working a hell of a lot in the past 20 years in faster methods, from 5 days to in a shift results. But if it still takes 48 hours to react, what's the point of paying more for a very fast method if you don't do anything with the results? So having in mind, not your time to results, but your time to decision, and digitalization does help a hell of a lot.
That's great quote. Time to decision, and you want the right information at the right time to be able to make the right decision, and making sure the right person.
Quote Martin, not me.
Yeah. Make sure about that. I don't want you to have any trouble with him. So yeah, that's, I think, part of the items we see here to help making decisions faster, so that it's over here, reduce time to decision. David, do you see any key benefit of digitalizing above, I mean, beyond what you mentioned earlier?
No, I think I pretty much covered it, Karim, around that. And that's a really good point. It's you got to have actionable data, and you got to make decisions about it. You've got to decide what action you're going to take, which obviously could be variable depending on the situation. So without the information, you can't get there. So no, I think I covered it already.
So all that is on the basically reacting according to risk that has been identified. I think one part that is pretty new and coming up now is on the predictive modeling, being able to identify coming contamination based on past results, which someone would do before based on his experience, knowing like you said, there's a crack here, I think there might be something there. So that's part of it. And there's probably a lot coming up now in predictive models. Actually, this is what we see at Novolyze as a very high potential way to anticipate contamination, and also to optimize the swabbing. So basically directing the swabbing where it makes the most sense and where you have the most chance to find a positive, and hence to be able to fix the issue. You know, AI and the latest technologies coming allow this to be a reality today, which is pretty cool, I find. Francois, any last word of wisdom on digital transformation?
It's important to use a digital tool that can soak the data from different sources. We are focusing today on processing environment monitoring. We still have this bad habit, in my understanding, that we trust the compliance on a batch on the finished product testing. I hope that this digitalization will help to have the complaints on a batch of all the information relevant to it, CCP under control for the different hazards, environmental monitoring under control for this period. So if you can gather information for your batch or for production on certain times from different sources, because we mentioned swabbing, but it's not all about swabbing. It's making sure that you didn't have any issue with the cleaning practices, that someone did not put a lot of water in the drying tower. You don't want that. Or there was some repair done, or there was an issue. David was mentioning that it's important to go on the field, like you have in Kaizen with Genba, having a look if something unusual happens. So all these aspects from GMO, from the different ways of monitoring the production system in the factory help for the root cause analysis when you have an issue with processing environment monitoring. So it's not only monitoring the processing environment, it's also monitoring the global data and be able to make the link between the different events, because at the end of the day, you need to explain what happened.
Right, thanks, Francois. We got plenty of questions here, so I'll start by a question from Sunny. The goal is that we want to find pathogens, but as time goes by, we fixed all possible root cause. Eventually, we won't find pathogens. Am I correct?
No.
That's a good question. It makes sense, and it's very practical. So
it's very practical. I think if everything was stable, I'd say yes. But in my experience, there is nothing in a processing environment that remains stable. The environment changes, the work changes, the culture changes, the business practice changes. You've got heavy equipment moving around, the floors get cracked, walls get dinged. So if one was dealing with a constant environment, I think you could say yes, but sadly we don't. So for that reason, this is a problem that is going to continue forever because the environment changes constantly.
Yeah, the factory is a living organism, living material with living bugs who likes to thrive there. François?
I will give you a return of experience from the drying tower in France following the 2017 Salmonella Agona and 2019 Puna outbreaks. The only way to get rid of a Salmonella-infused drying tower is to close the drying tower.
Right.
Basically. But we had some tower that went old, and we decided to close them, basically, because we could not get away with, we knew that it was too risky for the contamination. In the example of Puna in Spain or Agona in France, they went under the radar for many years, not seen by the factory, and suddenly, as David mentioned, you do some repair, you change some cracks, whatever is happening, you touch an old wall, you touch an old floor down the resin, and then guess what? You need to clean, you use some water, there is some organic matter present, and you end up with a contaminant. So no, you don't get rid of this. And I've noticed some factory with huge swabbing in place that did not find anything for 10 years. You've got an issue with milk powder on the floor, you need to clean very fast, someone is using water where he should not. Next week, you find back the salmonella you didn't find for 10 years. So no, you don't get rid of it, unfortunately. And it's the same point with Listeria. The first major outbreaks documented was with turkey sandwiches with Listeria monocytes 1-A, so that's the old serotypes, when we discovered afterward that it was in fact present for more than 15 years in the factory. No, you can have the bugs as far as possible from the production, but as soon as you stop the pressure, they come back.
Smart things. Ashley, can you move to the next slide, please? I think we have a very interesting topic here on the genetic characterization of these bugs. And you said, Francois, that there was this one that we found 15 years before. Was it through whole genome sequencing, or how did we find out that it was exactly the same?
Before, I mean, whole genome sequencing is definitely a game changer because it's reproducible and it's faster than PFG, another point, but it does not change epidemiology. And we knew about resident strains before using other techniques. I mean, the golden standard used to be PFGE. There was a solution with ribotyping, which did not go down to the strain level, but with elements from epidemiology. You know what was going on. It's very important to know the enemy. if you want to have a good strategy, know the enemy. We have discovered that for many years now that Salmonella has the capacity to survive in low moisture food environment. Listeria can survive in wet and chill. Why? And there is an interesting study done in Ireland when they make the link between resistance to basialconium in clinical isolates from Listeria monocytogenes, and the same in the environment, looking that we've been using quaternary ammonium compounds for many years in the food industry. And that's the point. If we have resident strain, we have a favorable ecology. So we need to understand what's favorable in the ecology in order to change it. Is it adherence to steel? Is it cleaning agents? Is it whatever? So what whole genome sequencing is not only making the link between food isolates and clinical isolates for epidemiology. That's the job in the US of the CDC, and it's very important, but it's also very insightful for the company when you have an isolate. Is it the usual pet? Is it something else? From the recent outbreaks we had last year in chocolate in Arlon for Ferrero or in Wiese for Barry Callebaut, they found Salmonella monophasic typhimurium. It was not the host pet, so they knew something was wrong with the ingredients. They found Tennessee in Wiese. It's not the host pet. They knew something was wrong. If you stop at the species, the area of action is very vast. So the characterization helps you focus really what's going on. Is it the ingredient? Is it against from the environment? What went wrong? Do I have issue with my cleaning practices? What does it survive? So the more you receive insights from your Chronobacter isolates, your Listeria isolates, or your Salmonella isolates, the more you can fine tune the control measures in order to change the ecology and make it less favorable for the survival of your house pet.
Interesting. David, do you have similar experience to share or other point of view on the impact of this genetic characterization?
Well, I agree wholeheartedly with what Francois said. WGS is a game changer. It really has changed it's changed a number of things. It's certainly changed the regulatory strategy here. And we have seen numerous examples where a regulatory agency has found a Listeria or a Salmonella strain from several years ago. It's an observation in a facility in a drain somewhere on a wall. It gets logged, it gets put in the gene tracker database, and there it sits until there is then an illness that matches it. And the regulators come right back into that facility, having identified a connection, and they do an infamous swabathon and they find it again, and they will conclude, unless you can prove otherwise, that it's been there ever since they originally found it. So you then end up with a window where they start to say, the regulators will say, well, we don't have confidence that you were controlling this for the whole time in between. And I think we've all seen examples. I certainly have, where that has triggered recalls that have encompassed everything in shelf life, unless the company has been able to show that we've had control of it and it's intermittent, which is difficult. So, Francois called it the house pet. That's a great term. And you don't want those. And you're not going to know unless you've leveraged some of this latest technology. That's a whole other webinar in and of itself, I think, Kareem, because the concerns and the fear around using whole genome sequencing by the industry is significant, because it's like, what if it gets subpoenaed? What if it gets discovered? That's not a situation that I think we've adequately resolved. But from a food safety perspective, it is a mighty powerful tool. And unfortunately, our regulatory environment has put the kibosh on us using it optimally. And we need to try to figure a way through that. But it's not being leveraged in the way it could be to protect public health and protect brands.
So I'll take your word on setting up another webinar on the topic. And thanks for that. And Ashley, how about we go to the next slide? We're going to have to wrap it up. We're already running out of time. So there is a quote from yourself here, actually. How about that? Any last word of wisdom, David? And I will ask you the same, Francois, so we'll wrap up this session.
I think that quote says it. Thank you for posting that. It is one of the greatest risks, and it's becoming a greater and greater risk, and we need to be on top of it.
Yeah, thanks, David. Francois,
I will make it shorter. Never assume things are under control, because the biggest killer in food safety is arrogance. So if you don't find something, search better, because there are always something to find. But what's very important is how this is promoted inside your company with the authorities, because you don't want to end up in a don't tell, don't ask approach. It's really being proactive, hurting yourself a little bit before real life hurts a lot. So that's really difficult training, easy war. So focus on the environment to avoid having to focus on your product.
Reminds me, very wise quote, reminds me of the quote of the late founder of Intel, only paranoid survive. Not far from that, a little bit more dramatic.
Not far from that, yeah.
Thank you. We have plenty of other questions. So what I'll do is that I'll follow up with you both so we can gather a couple of answers on those questions and share it with the audience, which was very active on the chat. Thank you again for participating to the webinar, Francois, David, and to all our participants.
Thank you.
All right.